Hello everyone, this is Mr. Friedhoff. I'm going to be going over chapter 5 in this video. And we're going to be looking at how cells transport things into and out of their membrane. So specifically, we're looking at how cells, again, just transport and move materials. And we're going to start off by just kind of reviewing what the membrane is. Remember that the membrane is composed of a bilayer or two layers of phospholipids. Now, there are other molecules within the membrane. You also have cholesterol and proteins. And these membrane proteins will help in that transition of molecules from one side to the other. But basically, this membrane just helps regulate what goes into and out of the cell. Now, remember, we have these things called phospholipids. Phospholipids have their head towards the outside and their tails towards the inside. These are going to be, the heads themselves are going to be hydrophilic because they are polar. The tails are going to be hydrophobic because they're nonpolar. So the outside head here likes water while the inside doesn't like water. We call that in science, it's called ampathetic. It's something not you don't have to know, but just a little added thing there where part of a molecule is hydrophilic and part of the molecule is hydrophobic. We also have these things called membrane proteins. These are within the phospholipid bilayer, and they have many functions. They can anchor cells together. They can work as enzymes. They can work as something called a receptor. They can be a transporter. But these items, these proteins, they're going to have a lot of different functions within the cell. Now, these membranes are going to help regulate what enters and leaves the cell. You can see here, these proteins can act as doors to the cell, like I said, but they're going to regulate what enters and what leaves the cell. We want to make sure we have the good things entering, and anything that's bad or not needed, we can expel from the cell. But these membranes and the membrane proteins are going to help facilitate that. All right, we have in section 2 something called semi-permeable. This is where some substances are allowed across, but not everything is allowed across. Think semi meaning some, and then permeable meaning it's able to cross the membrane or cross. This case here is, or this animation is showing how some molecules are going to move past the membrane, some molecules can't move through, and some molecules need a little bit of help to make it through or make it across the membrane. So what we're talking about here is how the membrane regulates what enters and leaves the cell. Some things can cross easily, while other things just need a little bit of energy. Now, in this section, we're talking about like polar and nonpolar things. Nonpolar molecules, remember, are molecules that don't like water. They can pass the membrane pretty easily, especially if they're small. Polar molecules, however, they don't pass the membrane very easily. They're not very permeable. So just a little note here, small nonpolar, they pass the membrane. You can see here hydrophobic nonpolar molecules, they pass very easily. Other small molecules can pass the membrane, but it's not as easily. But usually when they're polar, they're charged, they're large, they can't pass the membrane very easily. They need the addition of some energy to do so. Lastly, in this section, we're going to talk about something called concentration. This is the amount of something in a given area. You can see here where we have low concentration and high concentrations of something. So the more of a molecule or the more of a substance in a given area, we call it having a higher concentration. The lower the concentration, the lower of that solute or whatever. So again, low versus high, you can see high versus low up here. All right, now we're going to be talking about the specific type of passive transport. Our first type is called diffusion. This is the movement of molecules from areas of high to low concentration. This animation does a great job of showing us what that looks like. It's going to restart here in a second, but basically we start out with a high concentration of molecules here, and it's going to move equally out throughout this whole area. This is called diffusion. This is just basically molecules moving until they reach something called an equilibrium. At the end here, you can see they're going to reach something called an equilibrium. This is where all the molecules are about spaced out evenly throughout this area. Now, again, this is called diffusion. This is the movement. I'm going to go backwards here. This is the movement of molecules from areas of high concentration here to like low concentration here. And again, they'll reach equilibrium at some point. We also have something called facilitated diffusion. This is where proteins in the membrane are going to be used. You can see here we have molecules moving from areas of high to low concentration, and the only difference here is they're going through a membrane protein. Okay? Now we have osmosis. Osmosis is the movement of water. This is the big difference here. In passive transport, we're talking about, again, moving from high to low concentrations. Now we're going to focus on the movement of water. If you look at this diagram, you can see there are molecules and this solution. Now, it's in pink. I wish it was in blue, but look how the water here moves past the semi-permeable membrane. Do the molecules move? Do these little circles move? No, they don't move. what the water does. So you can see here what happens is the water adjusts because it's moving from the left side to the right side. It's going from areas of high concentration here to low concentrations here. If you look at our, again, animation, you can see this is water. The solute doesn't move past the semi-permeable membrane. The molecules of water do. This is a semi-permeable membrane. Water moves past. The molecules don't. Here's another way of looking at it. You can see osmosis here. The water is going to move past this semi-permeable membrane. However, these molecules here won't move past. So again, the water is moving past, but the molecules won't. They're moving from left to right, and you can see that water level change. Again, this does not require energy, and it occurs just naturally in nature. Now we have three different types of solutions. This is our last slide, and there's a lot on here, but we have hypertonic, hypotonic, and isotonic. These are just different types of solutions where in the hypotonic, there is more water on the outside than inside, so water is going to rush in. Isotonic, the solutions are equal on either side, so water is going to evenly exchange from side to side, so it's not going to change size. And then hypertonic. This is where water is going to leave the cell, so water is going to shrink because water is there's a higher concentration of water inside the cell than outside the cell. Okay, so these are the three different types of solutions we have to know for osmosis and passive transport. Now, in the next two sections, we're going to be looking at something called passive and active transport. I like this animation kind of showing us what passive and active transport kind of represents. We're going to learn that passive transport, this kayaker in black here, is going to be the example where it goes with energy or it goes with a flow. It doesn't require energy input. The other hiker here, or the other kayaker in color here, is going to be the active transport. It requires energy. If you think about this kayaker moving up the stream, it's moving up something called the concentration gradient. And this is going to require the input of energy. Where this kayaker doesn't need to add energy to move, this kayaker is going to need energy to move. Let's get into it. 5.3, we're going to be looking at passive transport. This is where we're going to go from high to low concentrations. You can see we're going from high to low. And this is just how naturally things in nature move. We move from areas of high to low concentration. It doesn't need energy. And there's three different types. We have diffusion, facilitated diffusion, and osmosis. So again, passive transport doesn't require energy and moves from areas of high to low concentration. All right, hello everyone. We're going to do a quick quiz on this content here. You can see we're going to go over hypo, iso, and hypertonic solutions. So looking at this one right here, you can see there's an equal exchange of water. That means that this one is a isotonic since water is going in and leaving and that cell remains the same size. This one right here, you can see water is leaving the cell. That means that the cell is going to shrink, which means this is a hypertonic solution. Here we see that water is entering, so that cell is getting bigger. That is going to be a hypotonic solution. If you look here, we have the cell, again, the water is leaving, the cell is shrinking. It is a hypertonic solution. This one, water is entering, so it actually burst because it just can't take on that much water. So this is a hypotonic. And lastly, we have that equal amount, so that's going to be an isotonic solution. Hello, everyone. This is going to be our last section in the cell transport chapter. This is going to be about active transport. So 5.4, active transport. So everything we just learned about passive transport pretty much just flip around. So this is the process of moving materials across a membrane, but we're going from low to high concentrations and we're requiring the input of energy. Remember that kayaker that had to expel energy, had to use energy to make its way up the river? This is the colored kayaker that's moving in the opposite direction of the river. And we have three different types. We have protein pumps, we have endo and exocytosis. These are pretty easy once you kind of look at them and see what's going on. Let's see what they look like. You can see in these protein pumps, it's just like facilitated diffusion, but in this case, we're using energy and we're moving concentrations up the gradient. So again, we're using energy or moving concentrations from low to high. This is that membrane. This is the membrane protein right here, and it's moving the molecules across it through the use of energy. We have endocytosis. This is where a molecule or molecules or just things are going to be taken up by the cell. And what happens is the cell wraps its membrane, forming a vesicle around it. You can see this is some liquid being taken in. These are some molecules. But this is basically endo or into the cell. This is how cells sometimes take things in. This is the opposite of exocytosis. This is where the cell is pushing things out. You can see that membrane, that vesicle, fusing with the plasma membrane, and then you can see those things that the cell doesn't need anymore being pushed out. But this is the opposite of endocytosis. Think exo, think exiting the cell. And this is our last portion of chapter 5. I wanted to show everyone this animation. This is a great animation, again, to show the difference between endocytosis and exocytosis. This first portion here is going to be endocytosis, where things are going to be taken up by the cell and they form a vesicle. The second portion here is going to be exocytosis, where things are going to be pushed out and that vesicle fuses with the membrane. So this animation shows both endocytosis here, coming into the cell, think endo meaning in, and exocytosis, think exiting, the things being pushed out of the cell.